a special thing for me today to talk to you. I'm going to talk to you about something completely different than what we're talking to until now. Let me just ask a question. How many of you are students? Okay. Do we have teachers? Some. No, not you. Okay, university professor. So I have my talk sort of designed for you guys, okay? So I think students is really where we want to talk to and get you guys inspired about life and about nature and science and how to study. So today our theme is understanding life, and so far it's been wonderful. We've been looking at all kinds of cellular mechanisms, and I'll maybe touch a little bit about that. But I'm going to talk to you about something completely different because I'm going to go on a limb and I'm going to talk about exploration. If this works. So let's see, here we are. All right, so I'm going to talk to you about cavefishes. And these animals are interesting because they live in extreme environments. I'm going to talk to you about things that I like to do in my lab, things I like to do with my students, and how life adapts and survives. And for some odd reason, as I'm getting older, the more interested I get into extreme environments. So that's me cave diving up there looking for fishes a while ago. So why are cave organisms interesting? So I'm showing you a bunch of animals, some of which we do study. If I had a pointer here, I would show you. So this is a cave crayfish that we do study in the lab. And you can see already that if you live in a cave, you have certain constraints, and you look sort of like each other, right? So you don't have big eyes, because there's no reason to have big eyes. You don't have eyes. And you also lose your pigmentation. You can see a lot of these animals are pretty much white or pale. They have nothing. These guys are some of my favorites. We have some amphipods up here, which we also study. All kinds of interesting invertebrates. Look at this cave spider here. Beautiful animal, very long arms and legs. And that seems to be one of the features or animals that live in caves. Cave is an extreme environment. If you think about it, the lack of light is a pretty difficult constraint to survive in, especially because most animals that we can think of live on the surface. Okay? So we use our visual systems to guide ourselves into what to do next. A lot of animals are visual animals. A lot of animals. Now, caves are wonderful experimental labs, natural experimental labs, because they're all over the place. And if you're lucky or unlucky enough to end up in one of these caves, you end up with very strong sensory constraints. How are you going to feel the environment? So today I'm going to not talk about a lot of these. I'm going to talk to you about cavefishes, because they not only have to be in a dark environment, which is tough on its own, but they live in a fluid environment. And if you think about it, dark water is pretty darn to survive. Okay, so I have a couple here. These are two animals. These are actually American cavefishes. They're from Kentucky and Tennessee. And sometimes when I go down south, I get this accent, but it's not my fault. And that's one of them here. Now, how do you survive in a cave when you're a fish like this? These are beautiful animals. I'm absolutely biased. The lack of eyes is the most beautiful, sexy thing I've ever seen in my life. Now, I want you to see other things too. They not only have these regressive features like lack of eyes, but they must have something else. They must find a way to navigate in the cold, dark water of a cave. And I have here, this is Astroblapus Cave. This guy is from Kentucky, and you can actually see his brain up here. But you will also see these little things on the side of its head. These are, well, on the side of its head. These are lateral lines, okay? These are mechanosensory organs. And the previous speaker was just talking to us about ear and your hair cells in your ears. That's exactly what it is. These are very, very long. Oh, thank you. Thank you, Michael. There's one up here. This is a lateral line. And there are a few up here. And any self-respecting fish has those. The thing about fish is that fish has a lot of them. Cavefish have a lot of these. So it's what we call a hypertrophy of a sensory organ. All right? So this is another fish. This is Asthenics mexicanos. This cavefish is from Mexico, and absolutely fun to go collect. And you can see here there's a lateral line. This is where the mechanosensory organs are. And just to point out and make things easier to see, I have stained them here. Well, I haven't, but I have them here stained with a fluorescent dye. You can see that they are all over the animal, and you're just going to have to take my word for it. This is a hypertrophy. There are lots and lots of these organs. This is what they look like. This is a scanning electron micrograph, so we can see them closely. And they're very long stereocilia. And what happens is, as they move around the water, or the water moves around them, they move. So whenever they basically stick up into the water, and they go like this, and they send an electrical signal to the brain. These fish are fantastic at creating something called a hydrodynamic image of their environment. So they can create in their brains an image of what their whole behavior, or what their environment is, so they can behave accordingly. Now, it seems that mechanosensory adaptations, such as this one, a hypertrophy of lateral lines, is a common theme in cave animals. And I set out to figure out if this is true. So one of the lines of research on my lab has been to look for animals that have mechanosensory adaptations. There's one caveat, though. If you look at a cave, this is a little tiny, small spring, or another cave, this is a big, quiet, long pool. Or if you look at a different season, this is sort of a spa cave. Or if you just keep looking even at something that doesn't have a lot of flow, you can have a small stream that's pretty fast. Okay? So not all cave streams are created equal. And that's something that people haven't thought about. You know, we just assume, okay, so you're in a cave animal, you sort of have tactile information. Well, it turns out that if you are in a cave which has a lot of flow, your ability to use your lateral lines, organs, gets a little bit constrained. It's sort of like you're trying to detect what your lover is saying in the rock concert. It's just not going to happen, right? It's way too low. The environment is way too loud. All right, okay then. So what do we do next? This is your best option. So I set out a few years ago in collaboration with some fellows and Natural History Museum in Ecuador, and we set out to study this animal. This is Estroblapus follitor. And I came into this project because, you know, here I am, the cave person, and I'm interested in going to cold, dark places, and I like animals that have mechanosensory adaptations. I expected this animal to have big, long neuromasts like every other fish I thought of. So that's how we started. So the museum and I looked at this animal, and I've been there 3 times already, and we started to try to understand how this animal lives, because one thing about Astroblapus is that it lives in this terrible place to live. It lives in a cave right up here on the edge of the Andes, on the east side as it comes to the Amazon basin. And it floods all the time. Their cave is torrential, maybe 4 times a week. That's a lot of water. That's a lot of water flow. What do these animals do? How can they use the lateral lines in a cave where current is so fast? Well, this is Astroblapus, and what Astroblapus does is it sticks to rocks. Not very surprising. If you had an algae eater in your fish tanks, well, that's basically what Astroblapus does. Astroblapus is about this big. This is an adult, and they are nothing but a big muscle with a sucker in the end. They are just so fast. They are so hard to catch. And they spend their lives stuck to rocks. And they go very fast swimming in the high current upstream to another rock, and they stick. They're actually very interesting animals. You can see the mouths over there. Another gorgeous creature, right? They have vestigial eyes. They're actually covered by skin. So it probably means to me that they haven't been in the cave that long compared to the American cavefish, okay, or to the Mexican cavefish. So these guys have been stuck to rocks in caves for, you know, a small amount of time, but long enough for the eyes to become fairly vestigial. So our first trip, we go, we find the animals, they collect them for the museum collections, and I say, oh, I will look at the lateral lines, because I'm the expert, right? And I don't find them. I don't find a single lateral line in this fish. And I have to say it was sort of a disappointment and embarrassing at the time because, you know, here I am supposed to be doing this and I can't find it. Turns out, and I'm going to go forward and backwards here, that if you look carefully at these animals, they have these little protrude. I don't have a good picture of this because this was taken before we understood anything about these fishes, right? But if you look carefully, there are these little sticks that stick out of their skin. So if you look here, there are a little bit of the sticks, and here's the eye. And if you do a scanning electron micrograph, well, voila, you see a lot of these things. And the moment I saw them, and I didn't see the lateral lines at the time, I thought, holy mackerel, what is this? And as a scientist, this is the best feeling in the world. I have to tell you, every once in a while, this is the second time in my career this has ever happened. It's the best thing there is. This is why people go into science. They go, wow, how can this be, right? So I looked at these, and I'll show you a close-up of one. And there it is. And it looks like a joystick. It's a bunch of joysticks that stick out of the skin. And if you section it, you end up finding out, I have some, we did various things. So we section it, and it turns out that they are teeth. They have teeth on their backs, and they're made of dentine and enamel, and you can see that if you do a lot of histology. So this is one of them. There's an innervation coming from the bottle. They are hollow, just like a tooth. It is a tooth. Bona fide tooth. And if you look at the bottom, you can see the nerves coming in. This is a transmission electron micrograph pseudo-colored, and you can see a nice myelinated nerve coming in and innervating the base. It is a tooth, which was pretty interesting to me. But I was here. I was in Ecuador, and I had to spend a long time thinking, I want to know why these teeth on his back, and why can't I find lateral lines? It was terrible. But eventually, with the museum, we were able to go back, and we're able to get a few more specimens, and we did some MRI. This is a MicroMRI scan, and we looked at the fishes themselves. We made three-dimensional reconstructions of the fishes, and we looked at their brains. And it turns out that these denticles, these teeth on their backs, were innervated by these gigantic nerves, which is wonderful. And they're here shown. This is an internal reconstruction of their brains. This is the brain. They're very large nerves that go towards the sucker, which is not a surprise. And you can see in yellow, these are the ears. Not to be a surprise either. But the important part that I want to show you are these two lateral nerves. And they innervate these denticles. And this is good. This is good for me because I could then go back with a specialized team, and we can actually record. Yes? They have a cerebellum? They have a cerebellum, yes. It's actually here. This is the midbrain, and the cerebellum is up there. Yeah. It's, no, they're not going. They're going to the brainstem. They're going to the lateral line. Listen, Ava stole my, my thunder here. These teeth, the innervation of these teeth, are going to the lateral line nucleus. Okay? And that is because these teeth, what you can do is you can go into it. This is my side picture here. Then I want to show you how it is like to actually do science in the field sometimes. This is our electrophysiological rig, which was done with a collaborator of mine who happens to be right here. in a chicken coop in Ecuador, and this is literally a chicken coop, and the ground, and Amy, who presented me, was there too. The ground was dirt, and we had moth, and we didn't have any lights, and it was fantastic. This is really the best way to exploratory science should be, right? You have to get moths and things, they kept falling into your prep, and it's just great. And what did we find? Well, I'll jump to the conclusion here because we don't have much time. These are electrophysiological recordings. And it turns out that if you are one of these denticles and you're sticking out of the fish's skin, and if you physically move them with a little pickle spritzer or piezoelectric, you can just move them. You send a neural signal to your brain. So what we did next, and I'm not going to show you for the sake of time, is we actually removed them when we put them on the fish into a current. And the fish had no ability to figure out what's up and down, what's left and right. These fish are using these denticles to create these hydrodynamic images that I was telling you about. So they're using teeth to explore the environment, to feel water flow, to figure out where they are and where there are things in the environment. Now, I told you in the beginning that I couldn't find lateral lines. Well, they're there. They're there, which is actually kind of a relief. But upon really more thorough, I had to really look hard. Neuromasts are there, the lateral line organs are there, and this is the comparative distribution. And what I'm going to show you here is one. This is a neuromast in one of these Astroblapus, and it looks small, and it looks pretty pathetic, really, compared to something like this. So this is from one of the other, actually this is from a guppy, you know, fish tank guppy. So you can see the difference, that something doesn't seem very useful is starting to go, or is small. Yeah, exactly. And it's probably not used very much compared to something that it's used a lot. You mean on the edges here? The water would, so the question was, is the water going to swirl into it? I'm not sure how that would work. Yeah, but I can bet you anything that they're less sensitive than an average guppy like lateral line. Okay, all right. So, so I wanted to. So this is a new sensory organ. I'm going to show you one last picture here. I want to finish up my summary because I know it's almost 5. So Astroblapus has a new way. This is a new sensory organ. Has a new way to explore the environment. Okay. So that's one of the things that I really wanted to emphasize here, especially for the students, that you honestly never know what you're going to find if you keep looking. Okay? And you just have to be aware and keep your eyes open because you don't know. You don't know where the answer is going to come. I was totally prepared to find my neuromasts, and I was totally prepared to follow my hypothesis, and it was just completely different. So, you know, science and nature show me the opposite way. Okay? So they have these neuromasts. So these skin teeth or denticles are much more abundant than neuromasts. They are connected to the nervous system. They're connected to the brainstem. They're actually connected to the lateral line part of the brain. So it's sort of like they've taken over. And without these skin teeth, without the denticles, the fish don't stick. So you can put them in a water flume, sort of a little artificial water flow. Actually, we almost lost a few in those. Amy almost lost a few in those. They will not stick. They swim. It's almost like, you know, I had a hypothesis that if you took half of them out, they will probably try to feel the environment with the other half. And it turns out that they will not stick to the surface. It's almost like they need that flow of information to be able to stick to the substrate. And so we went looking at other species. So we looked at some surface species because this is a large genus and there are lots of different Astroblapus out there. And it turns out that these skin teeth, these denticles, are present just a little bit around the mouth and on the barbs of the surface species, which is a beautiful thing if you think about evolution, because what it has happened is it has been an expansion, just like the lateral lines we've seen. There has been an expansion of these teeth in the surface species as an adaptation to the cave environment. So it's totally plausible and consistent and beautiful. All right, I wanted to spend 1 second thanking a lot of people. This is one of the teams that went to Ecuador. We go all over the place, including the US, which is easy. As a matter of fact, it's a surprise that I'm here and clean because I was in a cave not very long ago. And in Ecuador, because nothing is more rewarding for my work, at least, than to work with scientists and students from other countries. In my lab, we have collaborations with China. We have collaborations all over the world. And there's nothing more rewarding than working with students and getting students connected from University of Maryland, University of Shanghai, whatever, okay? International collaboration, it's been wonderful. So I am happy and delighted to take any questions. Yes? If I understand what you said, because I have a cold, I'm afraid, so my hearing is kind of lousy. So you expect animals from cave environments not to be related? Yeah. Yes! Oh, absolutely. That's exactly right. So cave animals are completely, and I showed you all these pictures, right? And that's the most beautiful thing about doing research in caves, is because it's a very extreme sensory environment. We'll get there. Whoops, that's me again. That's not good. This is it. These animals, okay, are unrelated. And all the cave fishes are unrelated. But they all undergo this extreme sensory constraint, and they go through this environment. So it's sort of like having the same experiment again and again and again, okay? So that's a wonderful thing, and you got it exactly right. That's why it's special. It's doing the same experiment with the animals in different kinds of orders. They're cat cave fishes, they're tetra cave fishes, they're, I don't know, loach cave fishes, which is one new species that we found in China a few years ago. Okay, so that's wonderful for biology. Good question. I don't know. So far, oh, I'm so sorry. So the cavefish that we found in China, for example, does it have, the same specialization as this one in Ecuador? And it doesn't. It has the same sensory specialization as the one in Mexico. Yeah, so definitely. Dan.
So the fish adhere to the surface with the current. Is it possible to measure how much force they're applying?
Yes.
So I guess they must grip stronger and bigger flow.
Oh, I'll bet. It's very, so if they're stuck, this happened once with me. He was stuck to a rock, and he did not want to let go of that rock, no matter what. So I picked him up, and he was just wiggle, but he did not let go of that rock, and I didn't. I couldn't get him off, so I collected him and the rock. Because what are you going to do? Yeah, absolutely. So let me just, I see you, but let me just, one more thing that I wanted to say, and that's relevant to the things that I do. So a lot of these animals are unusual, and they're beautiful, and they're rare. Even if we don't know if they're rare, they probably are. So nobody knows the population of this one in Ecuador. So one of the things that as a neuroscientist, right, my degree is neuroscience and cognitive science, completely unrelated to this, is conservation and being aware of the impact of the things that you do when you do your research. So, I don't have a C. elegans, which, they're lovely animals, and I think they're fantastic. Sorry, Eric, I do like them. They're good, yes. But they're not cavefish, so you don't have to worry about conservation issues. And it's something that I think as a scientist, and you guys as students, whatever you decide to do, it's outside the realm of your study, but it's something you should think about. What was your question?
Yes, in case where you found the fish with very few teeth around their mouth, would that be a case of evolutionary cycling?
Evolutionary recycling?
Cycling.
Cycling.
Maybe in that fish's history, we used to be a cave.
Well, one of the important things about, I tell you probably why not, and I'm putting my money on the not, is that because of all the ancestors of cave fishes, they're all surface fishes. Yeah. So, and then you have lots of different species where it is around the mouth and the barbs. So it's likely and more parsimonious that that state is the ancestral state, and that the derived state, the more specialized state, is the cavefish state. So that's where my money is. I think we need to go. Jean is giving me the eyeball. Thank you.